Gram staining procedure. This procedure was developed by scientist Christian Gram, and he separated bacteria into two groups on the basis of this procedure. This includes gram-positive bacteria and gram-negative bacteria. Steps of gram staining include: in the first step, we add a dye known as crystal violet. This crystal violet will stain both gram-positive and gram-negative to be blue. Then in the next step, we will add iodine. This iodine will form crystal violet iodine complex, and again both gram-positive and negative will be stained blue. It simply fixes the dye to the both gram-positive and negative bacteria cells. Then in the next step, we will add alcohol. Alcohol will decolorize the gram-negative. You can see they become white. So gram-negative, since they have thin peptidoglycan and they are lipid rich, so they will be decolorized. Whereas gram-positive have thick peptidoglycan, they resist the decolorization and they remain purple or blue. In the next step, we add a counterstain safranin, and gram-positive will remain purple or blue, whereas the gram-negative will take the pink color. So that's how gram staining works. So in the final step, gram-positive remain purple or blue and gram-negative become pink. So this is how, on the basis of gram staining, we have two types of bacteria, gram-positive and gram-negative bacteria. Gram-positive are stained purple or blue and gram-negative are stained pink. Gram staining procedure uses or advantages include identification of many bacteria, because it will divide the bacteria into gram-positive and gram-negative. So a large number of bacteria come under these categories. And secondly, it can help in choosing the choice of antibiotic, because gram-positive are more sensitive to penicillin. So this is how it helps in choosing the choice of antibiotic also. Limitations of gram staining procedure include that some bacteria cannot be identified by gram staining procedure also. For example, mycobacteria cannot be seen by gram staining. Treponema pallidum and other bacteria, it can also be not seen by gram staining. Remember, mycobacteria has a very rich lipid layer, so it cannot be gram stained. Treponema is very thin, so it can also not be gram stained, and there are some alternative procedures for seeing these bacteria. And then finally, another limitation of gram staining is that you need a large number of bacteria just to see a single gram-positive bacteria. That means almost 100k bacteria need to be present to see just one bacteria under oil immersion lens 100x. So that means that the procedure of gram staining has low sensitivity. If you have a readily available blood sample, you cannot directly do the gram staining. You have to first culture those bacteria, and that means that you have to give time for culturing the bacteria, and then you will see under the oil immersion lens. When you have sufficient number of bacteria, only then you can visualize a single gram staining bacteria. Now we can discuss the key differences between gram positive and gram negative bacteria. Here you can see plasma membrane. This is similar in both gram positive and gram negative bacteria. Just above the plasma membrane we have periplasmic space, which is present in both gram positive and gram negative bacteria. But remember, in gram negative bacteria, periplasmic space may contain some enzymes known as beta lactamases, which may degrade the antibiotic, for example penicillin. Then we have peptidoglycan layer, which you can see in gram negative bacteria it's thin, whereas peptidoglycan, which makes up the cell wall of bacteria, is thicker in gram positive bacteria. Then you can see the outer membrane and lipopolysaccharide. This lipopolysaccharide layer is only present in gram negative bacteria. It is not present in gram positive bacteria. And then finally we can see teichoic acid. These are fibers you can see, and these are present only in case of gram positive bacteria. Remember, teichoic acid are a feature of only gram positive bacteria, and they are not present in gram negative bacteria. And teichoic acid are made up of usually glycerol phosphate or ribitol phosphate. Both teichoic acid and lipopolysaccharide, they have a role in inducing inflammation, fever, hypotension. They have a role in giving shock-like feature when they cause infection in organisms. Now to summarize the key differences between gram-positive and gram-negative bacteria. Gram-positive bacteria, we said, have a thicker peptidoglycan as compared to gram-negative. Second difference was teichoic acid is present only in some gram-positive bacteria. It's never present in gram-negative bacteria. Then we said that lipopolysaccharide is present in outer membrane only in case of gram-negative bacteria, and it's not present in gram-positive bacteria. And remember, lipopolysaccharide has two components, lipid component and polysaccharide component. The lipid component has a role in endotoxin. It means it can act as endotoxin, and this means that it can act as a feature causing fever and hypotension. So this is a mechanism by which it causes inflammation and infection in humans. And then teichoic acid, it has a role in inducing inflammation and shock, just similar to lipopolysaccharide, which is present only in gram-negative. The teichoic acid, which is only present in gram-positive, has a role in inducing inflammation and shock.